Sure. Maybe I'll start off giving you a little bit of background about the University of Washington Virology Division as part of University of Washington Medicine. Um, we became aware of the outbreak in Wuhan very close to the beginning of the year when it was really an abstract issue in terms of U.S. health. And we decided at that point that we should begin preparation to have an assay available within the University of Washington system in the unlikely event that this virus came to the United States. And at the time, we did consider that an unlikely event. Nevertheless, we began test development at that time, which progressed through the month of January and February. The test was initially rolled out on a research basis because testing was centralized at the time at the Centers for Disease Control and later at state public health labs. Last Saturday, we received go-ahead from the Food and Drug Administration that academic labs such as ourselves could begin to offer clinical testing, and we began clinical testing within University of Washington Medicine Monday morning. The test is currently being run on a daily basis. The laboratory is now operating 24 hours a day with an approximate throughput of 1,000 samples per day. That is currently adequate to meet the demand, and we have plans to ramp that up to levels substantially beyond that should the demand require it. I want to introduce Alex Greninger, who actually led the team who developed this together with Dr. Arun Nalla within our group. And Alex can make a brief statement about the assay, and then we're happy to answer questions.
So the current assay we're using is the World Health Organization RT-PCR assay for detection of the COVID-19 virus, SARS coronavirus 2, to detect the RNA. It uses two targets to detect two different genes of the virus. Like Keith said, we're running tests daily here and with quite a high throughput. And we look forward to be able to contribute to control of this virus in this community.
I would like to make a point before we take questions that the University of Washington Virology program has a long history of developing tests within our program. These are called laboratory developed tests. They've been a critical part of patient care for over 20 years. In fact, our program developed some of the very earliest PCR-based tests for viruses like herpes viruses, cytomegalovirus, and many others that you may have heard of. Developing this test is nothing new for us, and we were very grateful when the change in policy from FDA allowed us to contribute our expertise to the management of this outbreak.
You mentioned that you can meet the demand right now, and if the demand increases, you can ramp up. What is the demand?
We've run about 200 samples, I think, in the first per day, the first couple of days. It seems to be growing. Remember, we've only announced the availability of our test recently, and clearly we don't know the trajectory of the outbreak right now in the local area, but we do expect demand to increase.
When do you anticipate that 1,000 tests a day would be a reality?
It's hard to predict the future course, but that could certainly happen within days, I think, and we're working very hard on our top priority, which is increasing our potential throughput in the lab.
Can you be clear about the specimens? When you say we can do 1,000 a day, is it the same protocol as the lab in Shoreline, where 2 specimens per person? So if we're saying 1,000 tests conducted, we're talking 500 people?
Alex can talk about the specimen requirements probably more detail than I can, but I will say that we want to move toward a single swab requirement from patients just to conserve reagents for the overall outbreak.
So right now we're taking nasopharyngeal NP swabs as well as oropharyngeal swabs. The nasopharyngeal swabs are probably the easiest to collect. We're actively working on development for sputum and BAL, which we should have soon for lower respiratory tract infections. But I've been in close contact with colleagues at the University of Nebraska Medical Center and Stanford University as well, who are performing testing and are looking at tests, and those are the best. Those have been consensus the best specimens to test so far.
We've heard from people that feel that they're exhibiting symptoms but feel frustrated that they don't have access to tests or don't need the burdens of testing. Will labs like this alleviate any of that or make testing more widely available to the general public?
I think it will help this, but as Susan pointed out, all this testing still needs to be ordered by a physician or other qualified healthcare provider. And really, much of the purpose for that is to be sure that the testing that's available does go to those who most need it. I know everyone would like to know, do I have the virus right now? There's simply not the capacity anywhere to do that kind of testing. But we do hope to meet all the need for people who are sick as they enter into the healthcare system.
Follow up to that, I don't know if this is within your ability to speak to, but can you talk to the burden of testing, and if you see that widening perhaps, and maybe the test becoming available to more people?
We're working with laboratories around the country to try to help them get their own tests up. I've been on the phone, Alex has as well, with colleagues. We are at the forefront here, and we do have a validated test that's available, so people are looking to us for help. We hope to assist in any way we can, especially in places that haven't seen the virus yet. If we can get them ahead of the game, that'll be wonderful for those areas.
I'd also like to say that the FDA regulation as well as CDC criteria have been actively changing over the last week. It's a very dynamic situation.
I was actually going to ask about it because just today CDC announced that those who qualify for testing, that has been loosened quite a bit.
Yes.
It's more up to the doctor now than it was in the past. What are your guys, just in the realm of this, what are your guys' thoughts on those loosened restrictions and how it relates to your research?
Well, I think it's great. I mean, it's useful not only from a research point of view, but simply the care of patients right now. That is, folks come in with lower respiratory symptoms and see a healthcare provider, that decision's made that testing is warranted, and now that can be freely done on the expertise of that healthcare provider. And that's where we need to go, not an abstract list of criteria. I think, as Alex said, this is a dynamic situation. Things are changing, and it's been wonderful that the regulatory agencies have shown great flexibility to change things as the epidemic moves along.
But also to that, one thing that I've sort of struggled to understand is why those federal agencies would keep it restricted to begin with. I mean, you mentioned, trying to make sure that people who need it are getting the available tests, but why would you restrict it in the first place? Why is that helpful in this process?
Well, I think that they restricted it in the first place because there was limiting testing capacity. And so it's basically between the testing capacity and the criteria, and to focus the testing on who needed the test the most. And those were known contacts of people who had been known to be infected, travel-associated cases, lower respiratory tract. And now, as we have more tests coming available online, we're able to offer a broader criteria.
I see. So the reason they can loosen the criteria is because folks like you are starting to develop more tests.
I believe that, and also just because people spoke up and pushed back and pressure. I think that's a large part of it.
You mentioned working with other labs. Are you working with other labs in Seattle and Washington State? Commercial labs, public labs? Can you say more?
We've spoken with a lot of labs all over. So here in Washington, we've spoken with private sector partners who have capacities that we may be able to leverage, but also laboratories throughout the country in other states.
And those private labs aren't currently authorized to do this kind of testing, is that right?
They're authorized in the sense that they can. They just have to validate the test and bring it up, and that does take some time. And we will work with anyone to get it down so we don't have to do any testing ever for this virus because it's never around.
Alex, can you talk about the development of the test, what that entails?
Sure. So like Keith said, we were interested. We began development of this as soon as the virus sequence became available in early January. And we had a number of tests that we have actually looked at over the time, and we've gone with what we have right now as the most analytically sensitive test, again, in collaboration with colleagues around the United States.
So the private sector labs are able to perform the tests, but they aren't valid unless...
They have to, under the current law is that board certified individuals running high complexity CLIA labs can offer a test once they have determined the performance characteristics and they determine them to be valid. That's basically what is sort of the letter of the law right now for testing for clinical care.
So is that the key to ramping up more testing, is for more commercial labs to be able to do it, and how do we get there?
Well, I think one thing is not everywhere, like Keith said, this is something we have a long history of doing, of making our own tests. There's actually legislation right now coming up in Washington called the VALID Act that sort of goes around this from the FDA looking at laboratory developed tests. And not all labs have that ability to make laboratory developed tests. And so what will also help the volume of testing is as manufacturers come online with tests that they can distribute around the U.S. and the world.
Is it fair to say that because you started researching that you're able to start testing sooner? Had you not done that?
Absolutely.
Yeah. Research, that's a very important point, is the research and the clinical care go hand in hand when we're dealing with these viruses. So the fact that we have a long history of research interest in how these viruses work, how they affect human beings, set us up very well to be ready to go as soon as we had the green light. Again, it was approximately 48 hours from the time the green light was given until we were up testing. And I really need to call out just the heroic work that our staff has done, and they're all upstairs working right now, but we had people here with us till 2 a.m. last night who came in again at 7 this morning to get these tests out to people, to make sure that they're accurate, they're efficient, and they're available. And we need to really, we owe them a gratitude.
Amen.
And when you say, and I think maybe you've answered this already, but I just want to make sure I'm clear. When you say you're working with other labs, do you give them sort of the keys to the castle, here's how you do it?
We're sharing everything. We're sharing everything. There's a huge spirit of collaboration here. This is a major health emergency. We all understand that. And no one is hoarding their keys right now. We tell everybody everything we know. This primer probe set works better than that in our hands. Is that what you're seeing? Yes. And here's what we're doing. Here are the specific concentrations of reagents in our assay. Here is the time that we take. All these things we're happy to share. You're seeing the same thing with the sequence information. You might have seen that the 3rd Washington sequence came out from our laboratory, and Alex and his group led that. This is telling us a lot about the transmission chains that are happening here in Washington State. We're working with our partner Trevor Bradford at Fred Hutch, who's really has a fascinating story about how this is spreading in Washington, really in real time. So everybody's speaking, everybody's bringing their expertise to bear, and nobody is hoarding anything at this point.
And one of the things that allows us to bring on these tests, what actually helps the development of the test, is access to positive specimens. And so right now, Seattle has more positive specimens. And so as Keith said, we want to send samples to other labs to help them bring on tests so they don't have to wait until the virus gets there.
Would we be in a, I know maybe I'm asking for crystal balls a little bit, but for the layman, I think a lot of us were wondering, why didn't we test sooner? Was it just the kits weren't available? Could we have been ahead of this, and as opposed to reacting to it?
In hindsight, there are things that could have happened to get this a little bit quicker, but this has been, in many ways, an amazing success story for the public health and the diagnostic arena. I know it doesn't seem like that because this is clearly a terrible problem that we're dealing with. But again, things are happening in a matter of sometimes hours, always days that would normally take weeks or months in our normal workflow. This is really a success story, and it is going to change the trajectory of this infection in the United States. So we're behind, in some ways, and we're hopefully ahead in others.
One of the short answer to your question too is that we weren't authorized to perform testing until Saturday.
But, and I'm sorry, just to clarify, I think you said this earlier, but just to clarify the timeline, you weren't authorized to perform testing, but this was already in development as of January.
Absolutely.
Yeah.
You have to be authorized. We had to be authorized before. You had to get an emergency use authorization from the FDA, and you can't become prepared overnight.
And the other thing we've heard over and over again makes a lot of sense is that the testing is going to be key. It's going to tell us a lot about this disease. So could both of you speak to significance of testing, the value of testing?
So testing is the main weapon we have against this virus right now. We can understand who has the virus, who doesn't. The people who are infected can be isolated from others, ideally at home, but in the medical system if need be. And social distancing can occur, and the things that have worked reasonably well in China as the epidemic has started to slow to some degree there. Really, until we have a vaccine or until we have clearly effective and widely available therapeutics, this is our major tool. So this is incredibly important, and again, that's why we're working around the clock to try to make as much available as possible.
And this virus has a long incubation time, and it has a long time until you get lower respiratory tract symptoms, and that's been one of the hardest parts about this virus for control, is that when we're seeing cases, we're seeing transmission events that could have occurred 2 weeks ago.
You mentioned that you've worked on developing tests for past viruses, some that have hit the U.S. and some that I imagine haven't. Can you explain maybe some of the differences or similarities with past outbreaks?
Sure. Maybe start, everybody knows about, hopefully, DNA and RNA. Some viruses are based on DNA, like we are. Other viruses are based on RNA, and how you diagnose those are completely different. The chemistry of everything, it actually has to vary. And then there's transmission. What does the virus manifest once somebody's infected? What's the sample that we need to test? It's critically important, and often that's a research thing we need to figure out. As a program, University of Washington Virology has worked for many years closely with clinicians, both within University of Washington Medicine and also the Fred Hutch, to understand how does testing fit in with the care of a given individual infected with a given virus. So we've done a lot of that work over many years. Those are questions that are open right now, and we're part of the teams that are trying to answer that for this new coronavirus as well.
Are you advising any commercial labs, working with any commercial labs, or aware of any academic labs in our state that are also?
I mean, we talk to commercial labs all the time, especially regarding what I said, specimens that are now positive specimens we have here in our laboratory. I'm not aware of other academic labs in the state of Washington, but I'm sure that some will come online.
Talked about the share of information. Has there been a share of information with the Chinese government or Chinese labs?
Most of that, I think, has been dealt with at the national level by CDC. There's been a lot of international cooperation as well. As Alex mentioned, aspects of our test are based on the World Health Organization test, and they've been very forthcoming with their information. So, yeah, we're very happy to speak with anyone, and that's happening around the world.
And there have been a lot of publications out of the 80,000 some odd cases in China. So there's been a lot of publications that have come out in the last month and a half, and that's been helpful. And then honestly, we get updates from colleagues and friends about what's going on, what people are saying on WeChat.
And then your take on how this is perhaps comparable to other outbreaks?
I think this one is extremely difficult. It's it's a respiratory pathogen with the incubation times that we talked about, and it has it seems to cause significant disease. Every virus is a little different, but this is one of the most challenging ones certainly I've seen in my lifetime.
You mentioned that you got approval from this a few days ago. What's unique about your testing compared to some of the others that have been done that it did require approval? What's the most unique thing? So,
all testing requires at some point approval by a regulatory agency. Up until Saturday, that approval needed to be done prospectively. That is, before you could offer tests, you needed to go through a very formal process with the Food and Drug Administration to demonstrate to their satisfaction that your test performed accurately and well. There's nothing inherently wrong with that conceptually, but the ability to do that in an emergency situation very quickly is simply slowed by all the things that are dealt with forms and applications and emails and delays and all the things that happen. The process now is essentially the approval is done retroactively, that when the lab, again, a high complexity lab that has been certified as capable of doing this in the past by other regulatory bodies. So, we have a proven reputation. These laboratories can do this validation, prepare those documents, but begin testing before there is approval. And in fact, those documents then have to be submitted within 2 weeks of the time the testing starts. And it's simply a reflection of the realization of how important this is to get this testing out as rapidly as possible.
And just to add one more thing, the testing, that method of testing, the current regulatory regime that we're under for this virus is the same as it is for every other virus. It was unique from February 4th until Saturday.
Do you know how many labs around the region
are currently conducting tests, and on any given day, how many tests are available in the Puget Sound?
In the region, I'm only aware of the state health lab and ourselves. From what we know of the capacity around the region, that's likely to be the case for a while. Clearly, we're looking for any avenue to increase throughput, and if there are partners who can work with us, we're happy to work with them and get testing going. We have no interest in controlling all the testing. We want to get as much possible for the people of the Puget Sound region, so I'm happy to work with anyone.
And how many tests are available, say, today, between you and the state?
Alex, what's the state lab?
So about, I'd say somewhere around 1,200 specimens.
So that would be about 200 at the state lab and about 1,000 here.
Let's say 1,200, a little over 1,000 individuals, patients.
And can you describe the symptoms or the criteria that you would need to meet for a doctor to give you the okay to get a test?
Well, so for right now, actually, it's up to the physician. And as we just heard, the CDC criteria are actively changing. Previously it was a lower respiratory tract disease that did not have a cause, but I think those have been significantly relaxed. And so really it comes down to a physician's decision for ordering the test. That's what we really could say.
Does it surprise you that the state lab has only tested 99 specimens in March?
No, the state lab is built for the purpose of understanding the epidemiology of transmission of viruses and other agents around the state, and they're incredibly good at that. We are built for providing large volumes of testing for clinical patients around Puget Sound and around the country. We do a large service for people from other parts of the country as well. So we are built to do a lot of tests, and surprising it is, we actually have more capacity in general than they do, simply because they, for the most part, don't do patient care. And that's appropriate, and that's why we partner, because we bring complementary areas of expertise to this.
You may be repeating yourself, Dr. Drummond. Would you mind telling me again how many tests you've run or specimens and the breakdown of specimens and people so far?
Right. So, Alex, I'll let you.
Sure. So we've had a slow rollout of the test, honestly, because we wanted to make it available to our hospitals, and before we sort of advertise it widely. That's why we're here today, honestly, to let people know that this is available. We've run approximately 200 specimens so far in the first 2 days, and I think today we'll see substantially more, and then tomorrow substantially more. So we're not anywhere near our capacity right now, and we are working actively to expand our capacity.
How long does it take to get test results?
So our goal is an 8-hour turnaround time.
In the first 2 days is
Monday and Tuesday.
Can you talk about the different sequences that you talked about and testing? How many sequences are now here in Washington?
I'm aware of 3 that have been deposited into the banks. We are currently sequencing multiple viruses that we've detected in our laboratory, and we have a pretty good workflow for that Alex developed along with Dr. Pavitra Rajchaudhuri in our group.
Yeah.
And those should be available possibly by the end of the day today. And those will be analyzed again by our colleagues, Dr. Bedford and others at Fred Hutch.
Yeah. So we've been sequencing excess respiratory virus genomes for the last 4 to 5 years here in the laboratory to understand transmission of viruses throughout the community as well. So it's something we've been working on for a long time, and now right now it's getting a lot more attention.
And just to be clear, you said that you guys have done 200 so far just since Monday. The state lab, I believe the number was in the 90 range. So already you have outpaced the state lab in terms of specimens that you guys are seeing the last few days.
I can't confirm how many specimens the state lab has tested right now. So if you're telling me that, then yes.
Apologize if it's already been asked and answered, but of this 200, are you giving out any information on the results?
Not in this venue, for health, protected health information reasons.
Do you have a sense of how quickly this virus is changing as it spreads and goes through different people?
So coronaviruses have very large RNA genomes. coronaviruses have very large RNA genomes. They're so large that they actually have to limit their evolution. And so this virus is changing, but thankfully, from a diagnostic standpoint, it has only recently emerged in humans. And so the levels of diversity that we see right now are very limited compared to other viruses. And so it makes that honestly a little easier to make a diagnostic assay for it.
Anything else you guys want to add or say that?
I just want to thank colleagues at the University of Nebraska, Jana Broadhurst down at Stanford, Ben Penske for talking with me a lot, Kirsten St. George at Wadsworth, New York Health, as well as honestly working closely with the FDA and the CDC in the last few weeks. They've actually been very responsive, and though we've kind of gotten a late start to dealing with this virus, they've updated their regulations and their thoughts over time, and that's a real strength.
And the governor's out. I'm sorry. Go ahead.
Well, I'd like to make 2 points. First of all, I want to thank everyone in this laboratory staff who's worked really tirelessly to make this possible, and I can't stress to you how much this has been accelerated over the usual development of a test. It's really a heroic effort on many, many people's parts, and we get to sit up here and sort of take the credit for that. But it is really a group of 60 people upstairs doing this work even as we speak. The second point I'd like to make is that we're talking a lot about potential volumes, and I'll say, we're talking about 1,000, and we've been looking at how we can get to do that. And in speaking with University of Washington Medicine leadership, they asked me what I thought we could do. And I said, with some effort, we might get as high as 4,000 to 5,000 a day. And to their credit, I was told by the highest levels of leadership within University of Washington Medicine that whatever resources we need will be available to push that to everything we can do. So I'm really grateful for that the University of Washington and University of Washington Medicine is really behind this in our efforts. And that support really means a lot because we want to do everything we can to stem the tide here.
I think it's interesting because it dovetails with the question I was going to ask, which is Governor Inslee made it clear that the lab in Shoreline needs more equipment. He was asking for more equipment. So does that mean that you're set with the equipment, what you have, your needs?
We can make use of more, absolutely. There is additional funding working its way through the United States government at the federal level right now. State support is incredibly welcome, and there have been feelers coming out from the governor's office and so forth. And then obviously anything we can do to help the state lab expand their capacity as well, we stand ready to do to return the favor of all the help they've given us.
And Seattle and the West Coast are the front lines for this virus. So we are what we can do to protect the rest of the United States.
I'm just curious.
Are you also working on a vaccine as well in tandem testing?
We are diagnostic testing. So that can help for vaccine trials. And we actually had one of the vaccine trials reach out. But we do diagnostic testing.
And I will say in the larger program, we work actively with vaccine manufacturers to help test their vaccines. We've been involved in many trials. Again, UW Virology has been involved in evaluation of vaccines for herpes viruses, for HIV, for hepatitis B. So as those trials come closer, we stand ready to help. But our first priority now is to meet the testing needs of individuals who may be infected.
And science aside, I mean, you guys were instrumental in these tests and possibly speeding up the system, which could have a huge impact positively on public health. How does it feel personally to know that you contributed to something positive with regard to coronavirus?
It's wonderful to be able to contribute. At the same time, I'm very humbled by this virus because we are not winning now against this virus. We've had a small victory that we have this testing available, but it's likely that the local epidemic is growing, and we don't know how big it's going to go. So our work is just beginning. By no means are we done.
One question for Alex, as someone who's worked in a lab setting, with this virus, has anything about looking into this sequencing, just looking at it, surprised you at all? As someone who works with viruses on a regular basis?
No, I think the one thing that's been unique is how recent it's emerged, and like I said before, how really honestly limited the genetic variation is right now. And so that's actually been very, very, very helpful.
And those are both positive things.
Yes, they are. The non-positive things are all the things the virus is doing. But from a sequencing standpoint, or a genome standpoint.
As this test is rolled out, are there any limitations geographically on who can get this test? Is it just Seattle for a while? Is it just Washington for a while?
So currently we are accepting specimens from throughout the country. And that is largely because we have capacity now relative to the local demand. That is extremely dynamic and that could change at any moment. And our first priority will be patients within the University of Washington Medicine system and local people. But at this point we are accepting specimens from outside.
I know you've touched on this already, just reiterate it and underscore, but people that are watching are here tonight who want to know, they hear that the lab is doing testing and they want to get the testing. How do they get it?
If they're healthy, they don't need to be tested, and the best thing you can do is wash their hands, stay out of crowds and stay well. If people are sick, a deep dry cough, high fever, probably above about 101, they should interact with a healthcare provider. They may or may not need to actually go to a clinic. There's increasing telehealth being implemented so that people don't have to be in large groups. But testing through our laboratory will only be available through healthcare providers, physicians or others. So, we can't see people independently. They can't mail anything in. They've got to go through the healthcare system.
How much does each test cost to test each specimen?
The test is priced right now a little over $200, which is generally in line with actually testing that we do for other respiratory viruses as well. So, yeah.
I actually just have a question from my newsroom about, and I don't know if you can answer this, when we should be using presumptive positive and when we should say a case has been confirmed. Are we still, the earlier guidance was that we should only say something's been confirmed if it was then sent to the CDC and confirmed? I don't know if that's something you can speak on at all.
Well, the volume, honestly, when I've been talking to people, sometimes hour by hour, there could have been new guidance. So I want to say, check the CDC webpage and the state of Washington public health webpage. But given the number of cases and everything that's happened before, and right now, our positive results and our inconclusive results, where one of the two targets is positive, tests, we detect it. All of those specimens go to the state lab, and where they're tested, and then I understand that those samples then also go to the CDC.
Thank you very much.
All right, great, thank you.